So I'm just gonna rewind since we didn't get too far into the lecture on this on Monday and just kind of review. So we're going through these invertebrate phyla just to review again what makes something an invertebrate. What does an invertebrate lack? Backbone, yeah, it's vertebrate, a vertebral column. What else? Doesn't have backbones, but it also doesn't have two other things that we mentioned. A cranium, yeah, which is what? Yeah, brain case. And then one more thing that most invertebrates lack has to do with the hard parts. Where are their skeletons if they have them? On the outside, yeah, usually. So you're talking about exoskeletons with one exception. So we'll get to that as we go through the groups. So we talked about porifera, which is your sponges. We talked a little bit about ctenophores, the weirdos, the cone jellies, and sort of their place on the tree. So we'll pick up with cnidarians, and we'll just keep on going through the list, looking at examples, looking at characteristics, talking about that kind of stuff. So remember, we looked at this relationship, and the big question here is who is basal, porifera or ctenophora? And we said the best evidence still supports that porifera is the basal lineage of animals. Not to say that the first animal was a sponge, but that's the oldest branching group, right? That there's a common ancestor of animals, and it looks like porifera branched off first. Okay, so that was the sort of take-home message about the ctenophores. They're an interesting group. They're really pretty. They're cool looking. And their phylogenetic history is interesting too. So that's where we sort of left off with those guys. So cnidarians, you'll need to know a little bit more about their characteristics. Remember with the ctenophores, I just kind of wanted to tell you the story. You don't really have to know a whole lot about them. I didn't give you a lot in terms of characteristics. Okay, just mostly the story that's interesting here is the phylogenetic mystery, right, that they're still working on. So we have some more characteristics to talk about with cnidarians. The cnidarians are named for cells called cnidocytes, and those cnidocytes are used for catching prey and for defense. So those are your stinging cells. So these guys are going to be things like jellies, coral is in this group, sea anemones are in this group, hydras are in this group. So you're talking about things that have stinging cells largely. There are some other characteristics that they have that are notable about this group. There's a lot of different body forms. This is one of those groups where you may frequently find sessile species. What does it mean to be sessile? They don't go anywhere. Yeah, they're not modal. So this is an anemone, and in sort of the cartoon version, and you can see that it's basically like got a stalk. It's like a root at the bottom. It's stuck to the substrate. It's not going anywhere. Its tentacles are up in the water, floating around like this. Its mouth parts are up here, and it's just bringing stuff into this gastrovascular cavity. So it's not going anywhere. But as a larva, it does. So its larval form can swim. Remember, that's what we said. If an animal is sessile, it at least at some point in its life had been modal, right? Because you have to be modal to be considered an animal. And then a jelly is kind of the flip of an anemone. So it's modal, kind of, right? It's mostly just going to be floating from place to place. They're not terribly active. But you can see a similar sort of body plan in these guys, which includes that radial symmetry. So remember, these guys are radially symmetric, which makes them a diploblast. What is a diploblast? What does that mean? You guys remember? What are we talking about? Yeah, two germline tissues, right? Endoderm and ectoderm. The mesoderm is lacking in radial areas, or radially symmetric animals. So they've got two germ layers. That's what's illustrated here in the yellow and in the blue. Okay, so what you're basically seeing is that internal layer and that external layer. They have a gastrovascular cavity. So when the blastocyst, that hollow ball of cells, starts to unfold and during gastrulation, that blastopore never goes all the way through. Okay, so you never get a full tube, a full digestive tract. It stops, and you end up with just one opening. So the blastopore persists as both mouth and anus. Okay, so in and out, and it's just a gastrovascular cavity for filter feed. So really simple. still less complex, right? Remember, the whole theme that we're sort of talking about as we go through the evolution of animal groups is that increasing complexity. Sort of what traits do you find appearing, what traits do you see disappearing, and sort of where do things arise in the phylogeny? Okay, so we're still looking at a pretty simple plan here. Let's see. Yeah, there's a little bit of detail in here about how cnidocytes actually work, but I'm not going to ask you about that, so we'll just skip it. Okay, so you don't need to know the physiology behind cnidocytes, but I do want you to know that those are the stinging cells that they use to catch their prey. That's important because that's what the group is named for, right? Or the cnidocytes. You guys good with this group? Fairly simple. Here's some pictures. There's a typical jelly caught a fish there, an anemone, coral, and that's a hydra. Just some diversity pictures to show you what they look like. Remember when we were talking about symbioses with protists, and we talked about coral and zooxanthellae? We mentioned it briefly. So this is just a close-up picture. I think you might have even looked at this picture before, of the coral polyp with the zooxanthellae inside. They're algae, they're photosynthetic chromalbulids. In the protist group, remember, they live in the tissues of the coral, and they photosynthesize, and they give that sugar to these coral. So these coral don't have to catch food. Okay? They depend on those photosynthetic zooxanthellae to feed them. But if they get stressed out, which they do pretty easily with changes in the environment, like temperature or acidity, they will eject those symbionts, which is essentially a self-destruct, right? Because if you kick out the protists that are feeding you, what are you going to do? You're not going to be able to get energy anymore. So that was just a sort of a revisit of that relationship. It's important because of symbiosis, right? It's important because we're talking about animals with protist symbionts. But it's also going to come up again when we talk about ecosystems later. We look at fragile coral reef ecosystems. So it doesn't hurt to touch on that again while we're talking about cnidarians. So that's going to get us to bilateral symmetry. Okay, so those groups that we've looked at so far, remember the sponges, mostly asymmetrical. Okay, ctenophores and cnidarians radially symmetric, multiple planes of symmetry. Right, you guys remember what that means, right? Symmetry is pretty straightforward. I think you guys got that. When we get to the next branch, that's where we see bilateria come in. So everybody beyond this point is going to display bilateral symmetry. What does it mean to be bilaterally symmetrical? How many planes of symmetry? One. You can only cut an animal with bilateral symmetry in half one way and still get two halves, okay, that have similar parts on both sides. So with the evolution of bilateral symmetry, we also see some other features come in. Okay, so these are going to be important. So we'll see this in all the groups going forward. Dorsal versus ventral also comes in. When you have left and right sides, you also have a top and a bottom. So dorsal means back side or top, and ventral means belly side or bottom. Okay, but it's easier to use terms like dorsal and ventral because if you have something with a back and a belly and it flips over, top and bottom is a relative term, right? So dorsal and ventral is an easier way to sort of describe something anatomically. Okay, whether you're talking about a lobster or you're talking about a human patient, those terms are going to be the same. So that's an important distinction to sort of make for yourself. So you know what dorsal and ventral means if you don't already. Right and left side, of course, if you're dividing something in half, down the center. Anterior and posterior ends, so head versus tail. Okay, that's again some terminology. If you're not familiar with it, now's a good time to start thinking in those terms because you'll hear that going forward, talking about all the animal groups, and if you're going into medicine at all, anterior and posterior, head versus tail. And then cephalization. So with a head end versus a tail end, you also start to get a head and the things that go along with a head, things like brains and eyes and eventually cranium. Okay, so those features go along with bilateral symmetry. The first group that we'll look at here are the Platyhelminthes, and these are the flatworms. Okay, there's 3 groups of worms, you guys, so I know it's very exciting, the differences between worms. They're going to be flat, or they're going to be round, or they're going to be segmented, and those are the differences. Okay, so hold on to your hats for that exciting discussion. The Platyhelminthes, there are lots of different examples. We're only going to look at a couple. We'll talk about planarians. Those are these guys. You might have seen these before in biolabs. So they're really easy to come by, they're cheap to order, they're cool laboratory animals, they're really, really small, but they're kind of cute. They have these little close together eye spots that kind of look like they're looking at you. But the really cool thing about planarians is that they have a tremendous capacity to regenerate. So I'm not going to make you guys watch this video in class, but you can look at it on your own. It's just a time-lapse video of taking one planarian and cutting it in half along this surface here. So right down the middle. So cutting it into a head and a tail. And within about a week, there's 2 new planarians. So this is cool in animals to be able to do that. So these guys, along with a couple of other examples like sea stars and axolotls, are of interest as model organisms in research because they can do this. They can regrow 2 new complete organisms when you cut them in half. And they do it really quickly, which is pretty cool. I mean, they're fairly simple as a flatworm. So it's not like growing a whole new person, but you know, it's still impressive. The genes to be able to do that and the cell signaling pathways that are involved, pretty cool. Tapeworms are in this group too. That's the next sort of notable species that we'll look at in the Platyhelminthes. That is the kind of worm that you hear about people getting as an intestinal parasite that latches onto your intestinal wall and just lives there and eats your food. So it's a straight up parasite. It doesn't, it's basically a big sucking mouth or scolex. So this is a close-up of its mouth parts. It attaches to the inside wall. Any mammal will do, including a human. And then its body is so flat that it can just absorb through diffusion your nutrients. Okay, so as you're eating, so is it. And it just grows longer and longer, and it grows in these weird little segments that are basically just full of gametes. So it's like a long, reproductive, parasitic eating sac. Okay, so that's what Taenia saginata is a parasitic tapeworm. You may not even know you have one. It may not cause you any problems at all, but sometimes they'll cause intestinal discomfort. Okay, but you don't really want one. Okay? Anyway, you guys have heard of tapeworms before, probably, right? Your pets may have had them. Yep. Okay. Yeah. So that's in this group. That's about it for flatworms. Planarians are cool. Tapeworms are gross. Moving on. Okay. Mollusca. That's a pretty big group. Lots of diversity in this group. They're known for their soft bodies. Okay, so you're looking at things like slugs, snails, octopi or octopuses are in this group. Oysters and clams, your bivalves, are in this group. They are, let's see, what do we want to talk about with mollusks? They have the soft body. Oh, lots of them have shells because they're soft body, right? So that for protection. The shell is considered to be an ancestral condition or an ancestral trait in this group. So mollusks that don't make shells now have evolutionarily lost them through time for whatever reason. Even things like octopuses, sometimes you will be able to find remnants of what used to be a shell, calcium carbonate that they still produce inside parts of the tissues. And then let's look at this. This is a really short video, and it's cute. It's really just for
I'm sure we'll talk about that.
Just for fun. But I like it because it sort of makes you think that maybe he misses his shell, in my opinion. Hang on a second.
Maybe he was testing those unique test situations.
He's got a coconut, you guys. 2 halves of coconut shell. They're also really smart. You guys know that about cephalopods, octopuses? They're very intelligent, problem solvers. Their vision is also really complex, like ours. Check them out. He's just gonna get in there. That was purposeful, don't you think? Like he picked it up, he carried it to where he wanted to be, and then just got in, like made himself a little home. That's it for mollusks. Questions? All you guys have to do for these groups, until we get to the really exciting evolutionary stuff, which is mostly happening in vertebrates, it's just characteristics and examples. Okay, so it'll be like matching or multiple choice type questions where you just have to pick the name of the group and you know who belongs there. So mollusks are things like bivalves, snails and slugs, octopuses and squids. Cool. All right. Annelids, one more kind of worm, or second type out of 3 worms that we'll talk about. These are your segmented worms. They're considered segmented because of these repeating body units. You can see them, right? Segments. Okay. The interesting thing evolutionarily or phylogenetically about this segmentation is that there are other groups of organisms that display segmentation, including arthropods, which are things like insects, crabs, spiders, things like that, and also vertebrates. So we're segmented. But there's a lot of work that's still going on to figure out if this is an ancestral trait that was lost in groups that are not segmented, or that we have convergently evolved segmentation in these 3 lineages, because they're fairly distantly related. This tree is showing where that segmentation came in, and so there's some work looking at the genes that lead to body segments, okay, and sort of how that evolution worked. I will not ask you any questions about that, but if you're interested in that type of research, there's a link to that article there. So if this stuff really like intrigues you, follow the little weird pathways that I give you guys. Otherwise, just know the examples in the group are things like earthworms. I don't have a picture of an earthworm up here, but you guys probably all know what an earthworm looks like, right? They're the ones that come out after the rain, and birds, robins come and pick them up, and they're on the sidewalk and stuff. Probably seen one digging in the dirt. Yeah, you guys know what earthworms are. Okay. Some interesting ones. Let's see. I've got a picture of a sandworm here. It's a type of polychaete. These are just kind of neat looking. They're mostly coastal, like saltwater species. They provide a lot of food for nesting wading birds at the beach. Leeches, that's an interesting one, right? They are bloodsuckers, so that's kind of cool. And then these are tube worms, which I always like to talk about because they kind of remind me of the whole coral zooxanthellae thing with the symbiotic relationship. So these, and they also don't really look like animals. They look more like plants, don't you guys think? Look like something you'd see growing out of the ocean floor or some other substrate. They grow near hydrothermal vents, so they live near hydrothermal vents, and they are somewhat sessile at this form. They live close to those vents where hydrogen sulfide is leaking out, right? We're talking about like volcanic gases and stuff coming out of these hydrothermal vents. They have bacteria that live in their tissues, really similarly to how the zooxanthellae live inside the coral tissues. And the bacteria that live in there are chemoautotrophs. Okay, so what does a chemoautotroph do? First of all, it's an autotroph. So what's it doing with as far as making food? Making its own food. But it's not a photoautotroph, so it's not using light energy to do it. It's a chemoautotroph. So what is it using? Chemical energy. So instead of using light to power that autotrophic synthesis of sugar molecules, these bacteria that live inside these tube worms use chemical energy. The chemical energy that they prefer to use is held in those hydrogen sulfide bonds, or the bonds in the hydrogen sulfide molecules. But they're not really good at getting a hold of that hydrogen sulfide, because as soon as that HS2 comes out, or H2S, sorry, comes out of the vent, it dissociates into the water. So it dissolves really quickly. But guess what? The tube worms are really good at sequestering that hydrogen sulfide before it dissociates. So they store it up in their tissues. They hand it over to those chemoautotrophic bacteria. They use the energy in those bonds to power not photosynthesis, but chemosynthesis. So very similar relationship to the photosynthetic animal symbiosis that we see in the zooxanthellae and the coral. But in this case, it's chemosynthesis, and it's in a hydrothermal vent. So just another cool example of symbiosis, endosymbiosis, right? Inside the tissues of another organism. So that's an interesting story. Probably the most interesting thing I can tell you about annelids, and segmented worms. Next group of worms, 3 of 3, the nematodes. Nematodes are roundworms. You may have heard of a couple of these species, particularly C. elegans. Anybody heard of C. elegans before? Nobody? Are you guys just quiet? So C. elegans is a popular nematode, sort of his name is bandied about a lot because it's a very important research animal. So C. elegans is a nematode. The genus is Ctenophora. So most people who work with it just call it C for short, right? C. elegans. It's the first animal that had its genome fully sequenced. So long before we knew the human genome, we knew the C. elegans genome. It's really important because using that information about their genome, we can learn things about our own, right? So we use them as model organisms to do genetic studies. We can also use them, they're really popular for developmental studies because we know exactly what every single one of their cells does from zygote to fully formed nematode. They reproduce pretty quickly. They can do it asexually. So you get a lot of animals in a short period of time and you can watch them develop from zygote to adult. It's really helpful because there's homology, right? These guys are animals. They're not that distantly related from us. They've got Hox genes, so we can use their genome and their cellular development to learn about how those Hox genes operate. It's a really important model organism, right up there with fruit flies, right, as far as being used in research all the time. So C. elegans is a notable species in the nematode phylum. There are two more that I want to mention. They both cause worm infections. They're worms, right? So they both cause like sort of parasitic or disease states in their hosts. Wuchereria bancrofti is the worm that causes elephantiasis. And I don't know if you guys have heard of this or not, but here's sort of a drawing of what happens in the patient with elephantiasis. I didn't put a real picture on here because they're kind of disturbing, but if you are interested, you can Google it and see what patients end up looking like. But what happens is these worms get into your lymphatic system. Do you guys know what your lymphatic system does? It's involved in your immune response for sure, because a lot of your white blood cells that actually fight infection take up residence in your lymphatic tissue, but it's also important in drainage. So your tissues are constantly being bathed and pushed with fluid from your bloodstream, carrying nutrients and dissolved gases to the tissue, right? But that tissue fluid has to be taken back up again, sort of reabsorbed and redistributed in circulation. So you have a whole system of vessels that runs along the same tracks as your blood vessels do that pick up fluid so that you don't just swell up and burst. Okay, so there's constant recycling of tissue fluid, just like there's constant cycling of blood. And these worms get into those vessels and into those ducts and plug them up. So they basically are growing in your vessel, in your lymphatic vessels, and blocking the flow of tissue fluid back out of the tissues. So you see it starts off with minor edema or swelling, and then you start to see it getting worse and worse, and eventually you even get like skin scaling because there's just no nutrient cycling going on. So that's elephantiasis. The scientific name is lymphatic filariasis. Lymphatic because those are the vessels that they're attacking, and filariasis because they're called filarial worms. So just know that that's a disease that affects humans that's caused by a nematode. This is considered a neglected tropical disease by the CDC, and I think we've mentioned that before, what that means. Basically, that this is not a big problem in the developed world. Okay? You're not likely to get filarial worms. They are also insect-borne. Okay, so places where insect control is more difficult, where you may need to sleep with a net, okay, because you may live in an open air shelter as opposed to a house where you can keep the bugs out, right? So it's a big problem, but not here, other places in the world. And then the other worm that I want to talk about, you're probably more familiar with than lymphatic filariasis because you might have to give your dog heartworm prevention medication. Anybody have a dog? None of y'all have dogs? I don't have one either, so no judgment, but usually it's like everyone has a dog. Have you ever heard about giving heartworm prevention medication to animals? Yeah, right? So that's to prevent infection with another type of nematode. You guys don't have to know these scientific names of these worms, just sort of know in general the diseases that they cause. So these guys, that's a picture of an infected dog heart with these particular nematodes that we call colloquially heartworms. They get into the heart and surrounding vascular tissue and clog circulation of blood. So you definitely don't want your dog to get heartworms because you can see healthy functional heart, no worms. This is not making a lot of room for blood flow, right? You're gonna see that. Worms all up in the heart. So heartworms are in this group too. So important research animals, C. elegans, particular tropical disease in humans, and heartworms in dogs. So those are the three sort of notable species in the nematodes. Okay, and that's roundworms. That's good. Platyhelminthes, flatworms, Annelida, segmented worms, Nematoda, roundworms. That's all the worms that we're going to talk about. Okay, I feel like the simplest, least exciting animal groups are behind us. That's just my opinion. So if you guys love nematodes, please don't take offense. Me telling you they're not that excited. But now we're getting into arthropods. This is getting cool. Okay, so we're talking about one of the most diverse groups of animals on the planet. Remember how we said 97% of all animal species are invertebrates? That's largely because there are so many species in the arthropod phylum. So arthropoda means joint feet. So pod or poda, think about like podiatrist. You have to go to the podiatrist, that's the foot doctor, right? So pod means foot, and think about arthritis. You guys know what arthritis is? You've heard of it before? It's pain or stiffness in what? Joints, yeah. So arthro means joints, poda means feet. They're named for their jointed appendages. If you look at this spider, you can see it. Their legs are jointed. Okay, so that's what arthropods are. They have an external exoskeleton composed mostly of chitin. Okay, we mentioned that when we were talking about fungi because they have chitin cell walls too. Almost 2/3 of all animal species are in arthropoda. So again, that's where that huge percentage comes from, and they're found everywhere. You find arthropods in all types of environments: aquatic, desert, the woods, wherever. Right? There's bugs everywhere, insects everywhere. There are some subgroups, subphyla, that I want you guys to be familiar with because this is such a huge group. We've got to subdivide it. The chelicerates, those are going to include horseshoe crabs, spiders, scorpions, and ticks. That's a horseshoe crab. You guys seen those before? It's the weirdo in the group because you can kind of imagine spiders, scorpions, ticks, they kind of look like they might be related to each other, right? At least in the same group. But horseshoe crabs are big. They're like this size or so, adults are. So it's kind of interesting that they're related to spiders and ticks. But those are going to be in your chelicerates. And then you've got crustaceans. So those are going to be things like crabs and lobsters and shrimp and barnacles. Daphnia. I think I have a picture of Daphnia in here. You guys may recognize. You recognize this guy? You guys didn't do the Daphnia lab in 1107 where you doused them in chemicals? You did, didn't you? Weren't you telling me, Cheyenne? No?
Brine shrimp? Brine shrimp.
Brine shrimp, that's what it was. With the same kind of experiment though, where you give them like caffeine and ethanol and stuff. Yeah, traditionally we've done an 1107 lab with these guys. You guys did do that? Did you take it online? Or some of you did, some of you didn't. Okay, so you remember now. These are tiny. This is a close-up, obviously, but this guy is a crustacean, so he's kind of closely related to something like a brine shrimp. Isopods, or what do we call these? You guys recognize this? Roly-poly, yeah. So that's in the same group, lobsters and crabs. So those are going to be your crustaceans. Then we've got Myriapoda. The myriapods are your millipedes and centipedes. I don't think I have any good pictures of those in here. Hexapods are your insects. If pods is feet, what is hexa? 6, yeah. How many legs do chelicerates have? Do you guys know? Spiders? 8, yeah. So if you can't tell what something is, count its legs. If you want to get close enough to see what it is. And then trilobites, this is like a fossil of a trilobite. These guys are notable because they were around for a really long time in the fossil record. They disappeared at the KPG, which is the same extinction event that got rid of the large dinosaurs, all the dinosaurs except birds. So these guys were historically everywhere, and then they didn't make it through that particular extinction event. So those are your subgroups. Let's see. Other than looking at some pictures and being able to put them in the groups together, that's pretty much all you need to do. Here's some photos of some insects. Insects are super diverse, right? Half of animal species are insects. So 2/3 of all animal species are arthropods, and half of all animals fall into this subgroup of arthropods, the hexapoda. So you've got stuff in here like butterflies and moths and bees and what else? Beetles, ladybugs, stuff like that. All kinds of examples of insects, mosquitoes, dragonflies, fruit flies. I mean, the list goes on and on, right? Cool, so that's arthropods. Another group, check. You guys doing okay? We're moving through the list. Now we're getting close to home. We have made it to the Deuterostomes. You guys remember what it means to be a Deuterostome? Yes, mouth forms second mouth. So anus forms first. When the blastocyst starts to gastrulate and the blastophore starts to fold in, that first hole is going to become, that's going to be the anus. The second hole to form will be the mouth, so Deuterostome. It's just that developmental pathway that puts this group in with the chordates, which includes us. So we're getting closer to, we're sort of narrowing down. We'll end with humans. Okay, so we're going to just keep on heading in that direction as far as going through these groups. So here we are with Echinoderms or Echinodermata. Okay, that's the full name of the group. These are going to include things like your sea stars, sea urchins, sea cucumbers. Have you guys ever seen a sea cucumber? These things, if you see them like laying on the beach, throw it back in. But they kind of look like those, you guys may or may not know what I'm talking about. It's a toy that you can get at the store. It's full of water. It's like a tube within a tube. They look like, yeah, I know what I'm talking about. And you can do like this, you know what I'm talking about? And just like slip through your hands. That's what they look like. They're super weird. But it's interesting to think about this as a close relative to chordates, right? So echinoderms are named for their spiny skin. That's what echinoderm means. And let's see. Oh, this is an important point. They sort of look radial. Didn't we say that everybody beyond the point of the split with Cnidarians were bilaterally symmetric? We did, right? So these guys would kind of look radial. They're not. If you take them apart internally, they're not radially symmetric. But you can really see it in their larval stages. So they go through multiple phases before they become the sea star that you know and love, or these other guys. And those larvae have that bilateral symmetry. So it does exist. It's just sort of a body form thing that they end up looking kind of radial. These guys are also notable for this last point here, that their spiny skin covers a skeleton of hard calcium carbonate plates. What is unique about that? It's not necessarily the calcium carbonate, and you see that in shells. It's on the inside. Yeah, so this is the exception. When I said most invertebrates lack an endoskeleton, these dudes have endoskeletal elements. Okay, it's not the same as yours. It's not the same type of tissue as your bone. It's not made of the same stuff, but it's not too far off either. It's calcium carbonate. Okay, so they've got hard parts inside the skin on the outside. That is an interesting shared characteristic as a deuterostome, right? That's notable, I think. You guys want to see a headless chicken monster? Want to see what that is? Who's got time? Who doesn't have time for a headless chicken monster? This is just when I don't want when I want the ads to play, they don't play. Hold on. Okay. Yeah, this is an example of a particular species of echinoderm, fondly called the headless chicken monster. It's a sea cucumber. Or sea, yeah.
Looks like maybe Nancy's to me. Maybe Nancy's to yeah. Oh, that's beautiful.
These tentacles, which are modified tooth feet, scoop in the sediment, and they are only taking in material from the surface of the sediment, which is higher in organic material.
But they do spend a large portion of their life in the water column, whether that is to get somewhere much faster or it is just swimming at all. And that's something that these polarvaries are known to do. They drop as much weight as they can just before they lift off. But hey, see, it's either trying to get somewhere quickly or more likely trying to evade a predator.
Yeah, it looks like a chicken on the side. You guys see that?
The name Enipneastes means dreamer. So dreamer is certainly an apt name for the way that sea cucumbers swim. Carney tells us that in addition to the name dreamer, common names include the Spanish dancer and apparently a favorite among Gulf of Mexico industry ROV pilots, the headless chicken monster.
My story makes no sense. What going on? There's like a
Yeah, let's check in.
I feel like it looks like I have a stick. I see it. I don't know, that lady wasn't feeling it, but I get it. So that's a weird sea cucumber. They're benthic, like she said, they live on the bottom, but they can lift off and go in search of food and stuff like that. Kind of an interesting one to take a look at. I feel like if I teach you biodiversity and I don't tell you about the headless chicken monster, I'm doing something wrong. All right, let's talk about chordates. We're getting really close to home now. Okay, so this stuff, we're changing gears a little bit. These next two slides are talking about the key characteristics that separate the chordates from all the other animals. This is important because these are traits that you share with your closest relatives in the animal groups. And chordates is the phylum in which we belong, right? So you are a chordate. So we're starting to get into, we're getting really close to talking about vertebrates. And we're starting to look at the traits that, these really complex traits that evolved relatively recently, within the last 500 million years or so, give or take 100 million years of animal evolution. So yeah, in geologic time, that's not that long. Okay, so all of these things we're talking about new derived traits that lead to complexity. Okay, that eventually we're gonna get to us on the tree. So the four traits that unite chordates are the notochord, okay, which is this right here, this structure. It is made of like almost like cartilage. It's firm, it's stiff, but it's also flexible. So it's going to allow support on the longitudinal axis. Okay, so you're starting to get towards that the head-tail organization, but with that flexible support rod. Okay, that's going to give rise to some important things later. You have the dorsal hollow nerve cord. Okay, what does dorsal mean again? Top or what? Back. Yep. So it's on the back side of the animal's body. Other organisms that are not chordates also have these nerve cords, but they're not dorsally located. Okay, so they're more likely to be ventral. So that dorsal location is unique. It's also hollow. So it's not empty. It's usually fluid filled. In us, it's cerebrospinal fluid. Okay, so we're talking about in us, your spinal cord. So a dorsal hollow nerve cord is a shared trait among all chordates. Pharyngeal gill slits. Do you guys know what a pharynx is? Or have you ever been diagnosed with pharyngitis? Any medical folks in the room? Pharynx means throat. Okay, so pharyngitis is just a sore throat or inflammation. Pharyngeal slits are actual openings in your pharynx region, in your throat region. So fish, you can still see these, right? Aquatic organisms that breathe through gills, these are gill slits. Okay, these are going to be the arches that support those gill openings. When you were an embryo, you had them. Okay, you had gill slits, you had these arch supports. As you develop towards birth, they give rise to other things. So they don't stay gill slits because you don't have gills. But you do have things like jaw bones, ear bones that help you with hearing, right? Internal facial structures, those all come from those pharyngeal slits. So they change and they morph as your embryo is developing, but you had them at one point. Okay, and then the same thing with the tail. So the muscular postanal tail. That means your tail goes beyond the end of your digestive tract and it has muscular elements to it. Do you guys have a tail? You don't, but guess what? You did. You used to have a tail. Usually, in most cases, with human babies, it's reduced by the time you're born. Great apes too, like chimpanzees and bonobos, they lose their tail. But you had one, okay? So that is a critical part of this discussion. All chordates display these four characteristics, at least at some point. So although some groups, the traits may only be present during embryonic development, but you had them at one point. It's kind of like the motility thing, right? All animals are motile, even when they don't move, did at some point in their life cycle. Same deal with these characteristics of chordates. So we'll look at a whole bunch of groups of chordates. We're going to go through all the vertebrate groups, the familiar stuff like birds and mammals and reptiles. But there will be a couple of groups along the way that are less recognizable. Okay, but they're still chordates, so they have these traits. They may just look a little different because they may have them and then lose them, or something along those lines, okay? So these are the same things we just talked about, but now you've got the words. So you got picture here, you got words here to describe those 4 characteristics. So you'll see multiple questions about these. Because it's important since we're switching over to starting to talk about vertebrates. That's it for the invertebrate chapter. How do you feel about animals without backbones? You guys are okay. Some of you are nodding. You're like, they're all right. 97% of animals, right? Are you psyched about vertebrates? Okay. Well, we've got about half an hour, so we'll spend some time talking about vertebrates. Luckily, this is getting more and more familiar, right? I hope. Okay. Okay. Characteristics. That's how we kick it off, right? All vertebrates are members of the phylum Chordata. That's what we just talked about. Okay, but there are going to be chordates that are not vertebrates. So the first part of this chapter, we're transitioning from invertebrates to vertebrates. There's going to be that weird transitional group that is a chordate, but not a vertebrate. Okay, so we're going to talk about the differences and sort of how you get to true vertebrates. So within chordata, you've got a couple of weirdos on the outside. Then you've got craniates. So we'll look at these 2 invertebrate chordate groups are the weirdos. We'll look at those first. The next group in line are going to be considered craniates, but not true vertebrates. Okay, so they have the cranium. Those cranial elements start to come in. Either a bony brain case, a brain case made of cartilage, so the word cartilaginous means it's made of cartilage, or fibrous. So it may not even be cartilage, it may just be sheets, like connective tissue sheets, but you're still getting that element of something to protect your developing neural central. Okay, so craniates are going to have that bony, cartilaginous, or fibrous structure that surrounds the brain, and may include a jaw and facial bones as we move forward into the more derived groups. Then vertebrata is the next clade. That's your true vertebrates. These are going to be the ones that have that true vertebral column, like we talked about, composed of individual vertebrae. Remember, we said it's not one particular bone. A backbone is not a solid bone, but rather a collection of vertebrae that articulate together, right? So that's what we're talking about with our vertebrata clade, is that true backbone or collection of vertebrae. 62,000 or so species identified, including those that are extant and extinct. Here's an interesting statistic that you'll probably hear about. Lots of threatened vertebrates. When we talk about endangered species or threatened species, there are quite a few on the list that are vertebrates. 29% at this point of amphibians are threatened. Do you guys remember when we talked about chytrid fungi and chytridiomycosis? It's been a while and you already took a test on that, so forgive you if you don't remember, but it causes that skin disease in amphibians. So that's one of the reasons for the decline in amphibian diversity, is that chytridiomycosis, but also habitat loss, because a lot of them live in tropical places like the rainforest, right, which we tend to deforest to do things like make wood and plant coffee and stuff like that, right? And 21% of all mammals are endangered. That's kind of interesting to think about. Why do you think mammals are not endangered? Sorry, threatened. So endangered includes threatened, but we're talking about threatened here. So a fifth of all mammal species face some sort of threat from humans. Why do you think that is? Any guesses? Why are any animals threatened? What do we do? Pollution. Say again? Urbanization, habitat loss, right? Mammals tend to be, so we are mammals too, right? So we tend to share niches ecologically. We tend to share resources like space and food and water, right? And so as we develop more and more habitat, it's more likely that we're putting pressure on other mammal species. Also, a lot of them are big, right? So if you need space, like an elephant, and you're competing for space with people who want to build ranches and things like that, you can see where the challenge might come in. So that's sort of an interesting thing to know. When we start talking about conservation biology, we're often going to be talking about vertebrates for those reasons. All right, review. We just did this, so I'm not going to do it again. Okay, if we had done this in a previous lecture, I'd go right back over it with you again, and we'll review it later, but we won't do it now since we just did it 5 minutes ago. 4 key chordate characteristics, okay? So just keep those in mind. That's exactly the same slide we just saw. All right, let's talk about the invertebrate chordates, and then we'll get into craniates, and then we'll get into true vertebrates. And it won't take long because there aren't very many we're going to talk about. There are 2 groups of invertebrate chordates. These are organisms, groups of organisms that have those 4 chordate characteristics: notochord, dorsal hollow nerve cord, pharyngeal slits, and a tail. Okay, at least at some point. But they don't have a cranium, and they don't have vertebrae. Okay, so they have those traits that make you a chordate, but none of the stuff that makes you a true vertebrate. You've got your Urochordata, which are also called tunicates. You may have heard of tunicates or sea squirts. They're sometimes called sea squirts. Because they have an incurrent and excurrent siphon. So these are tunicates. It's kind of hard to see, but what basically happens with these guys, here is a larval tunicate, okay? Or a schematic of a larval tunicate. You can see it has pharyngeal slits, it has a dorsal hollow nerve cord, it has a postanal tail, it's got the notochord. So it's got all 4 chordate characteristics. But larvae swim for a few days, attach to a suitable substrate, and then undergo metamorphosis to turn into something that looks like this. So this is a larval tunicate, and that's what an adult looks like. And that's a picture of what the adults look like, at least this particular species. They're not all blue, but the blue one looked cool, so I chose that picture. When it metamorphoses, it only retains pharyngeal slits. So the only chordate characteristic that it has as an adult are those throat slits. Okay, everything else goes away during metamorphosis, but they persist as part of this filter feeding sort of setup. Incurrent siphon here, excurrent siphon here. So mouth and anus still present, right? But water comes in, water goes out, and they squirt out those sides. So in this one, this is arranged a little bit differently, but here's incurrent siphon or mouth, there's excurrent siphon or anus. So water comes in and water squirts out. That's why they call it sea squirts. Okay, so those are the urochordates. Hard to believe those are so closely related to us, right? Because they're so such weirdos. The other group of invertebrate chordates are the cephalochordates, or the landslets. So these guys are starting, at the very least, starting to kind of take on an appearance that's recognizable as a vertebrate. They kind of look like what, do you guys think? Fish. It's kind of like a minnow, right? So that's the shape that they have. They are largely benthic, meaning they bury down in the substrate at the bottom of the water, and that's how they live. But they have all 4 chordate characteristics, and they retain them as adults. Let's see, what's mostly interesting about these guys? They're just landslets. They don't have a cranium, right? No cranial elements. They don't have vertebrae, but they have all those 4 chordate characteristics, and they retain them. So they're not super exciting, the landslets, but what's interesting is looking at relationships, right? If you're into that kind of thing, which clearly we are in this class, right? Looking at phylogenetics, the burning question in animal phylogeny, chordate phylogeny, is who are more closely related to modern vertebrates, urochordates or cephalochordates? If you had to guess, what would you say? Cephalochordates, right? Why would you guess that?
The only thing they're really lacking is the head cavity. Pretty much the vertebrae.
Yeah, they just don't have vertebrae and cranium, but they look like they have a head in, right? They don't really have eyes, they don't have a brain yet, but they have that sort of structure, and they retain those 4 characteristics. So that's been the subject of research that's been going on for quite a while, in the not too distant past. And most molecular systematics tells us that we are more closely related to cephalochordates than we are to urochordates. So the hypothesis is supported based on just morphology. When you do molecular work, the same relationship comes out. But interestingly, in the late 1990s, there was a discovery of these fossils called Hycuella that are a transitional species between cephalochordate ancestors and modern vertebrates. So Haikouella is cool because although he still does not have a cranium or vertebrae, there is a brain and eyes. So he's more complex, he or she, Haikouella. They're more complex than modern lancelets even. So they're extinct. So we don't know what there's none of them around to look at live ones, right? But we have lots and lots of fossils. Actually, a huge collection of fossils has come out of China with these guys. And they provide fossil evidence to add to the molecular evidence and the morphological evidence looking at modern day lancelets that cephalochordates are more closely related to modern vertebrates than Untichordates would be. Okay, so this is an interesting guy just because transitional species are always fun. Always important to find when you're looking for evidence of evolution and how groups transition from one thing to another thing. Sort of like the missing link, right? So that's an interesting species there to take a look at. All right, on to vertebrates. You guys ready? We've got through the weird invertebrate chordates. Then this is your list that is matching to the sort of list that we have in the invertebrates, where I'm going to give you the groups that we're going to cover, and then we'll talk about them one at a time. Okay, there's going to be a little bit more to this discussion than there was to some of the invertebrate groups, just because we're talking about evolution of derived features like jaws, appendages, amniotic egg, hair, stuff that we're sort of getting to closer to what we have. All right, so agnathans are jawless fishes. We'll cover those guys first. Chondrichthyes are fishes who have cartilage skeletons. Osteichthyes, bony fish. Amphibians, reptiles, which includes birds. Birds are reptiles. You guys may or may not know that, but now you do. And then finally mammals, including primates and us. Okay. All right. Let's look at the tree. We'll keep coming back to this tree, right? Here are the traits that we're going to trace as we go through these groups. We're doing great. Okay. I did put the little box in here that says this relationship is debated, but that recent molecular evidence supports cephalochordates. So this tree, what does this tree tell us about that relationship? I'm gonna let you guys tell me. Why did I put this box here? Anybody feel like reading trees this morning? Whoops, all right. Let's go through it together. Here we have the ancestral deuterostome, and you got a node here that splits into echinoderms, which are still invertebrates, right? And then the next node here is going to give us cephalochordates, and then the next node, urochordates, and then the rest of the animals here. So this, what does this relationship look like on this tree? Yes, so Cheyenne is telling me that the urochordates appear to be more closely related to the rest of the animals than the cephalochordates on this tree. You guys see that? If you were to make a monophyletic group and you went here, you would include urochordates, but cephalochordates are on the outside. You guys see that? So they share a more distant common ancestor with the rest of the animals than the urochordates do. So this is the tree that's in your book. But remember, we just talked about this, that it actually should be based on molecular systematic data and the presence of Haikouella in the fossil record, the flip, right? But that's still debated. So it depends on who drew the tree. Okay, so this is a really good illustration of how phylogenetic trees are hypotheses. Okay, so this is, maybe it's right, maybe it's wrong, maybe there's more to learn still, but just know that there's still a question mark there. Okay. You guys love phylogenetic trees, don't you? I can tell. I get so excited when I start talking about debated phylogenies. I'm fired up. Okay, I'm joking, clearly. You guys also love my jokes, I can tell. Okay. So we're looking at the chordate ancestor here. Craniates come in here. Vertebrae, we'll look at the introduction of those, where they truly evolved. The jaw. Okay, the jaw is a huge innovation in animal history. We'll talk about the advantages of having a jaw. You can start thinking ahead. What can you do if you have a jaw? Lungs, that's a big deal, especially for terrestrial vertebrates, right? Those of us who live on the land. 4 limbs, that's a big one. legs on land, amniotic egg, okay, reproduction on land, so we'll talk about that, and then milk for mammals. So those are some really important evolutionary innovations that are going to appear as we go through these groups. I told you it was getting super exciting. All right, let's look at the Agnatha. So this root word, G-N-A-T-H, nath, means jaw. What does it mean when you see a or an without? So agnathans have no jaw. They're jawless fishes. So the first vertebrates evolved in the ocean. Okay, so the first groups that we're going to look at are all fishes. So the agnathans are the earliest of the fishes that you see any extant examples of today. Okay, so there are older lineages than these examples that we're going to take a look at, but we're focusing on the extant ones. Okay, so the ones that are extinct, we're not even really talking about too much. I may have a picture of Dunkleosteus. We'll look at him in a second. He's extinct, but cool. Anyway, these are hagfish, and these are lampreys. Not this guy, that's a real fish. This is a lamprey stuck to the outside of that fish. Okay, this is a lamprey, that's a lamprey's mouth. All right, so we're talking about hagfish and lampreys. This is also kind of a debated group as far as are they or aren't they vertebrates. Now, hagfishes are generally not considered vertebrates. So these guys would be craniates, but not true vertebrates. Okay, so they have a cranium and they have eyes, although their eyes are fairly simple compared to ours. They do not have jaws. They do not have paired fins. They look kind of like a tube. Okay, they have their notochord retained as adults. They are famous for their slime glands. Oh, we got time. Let's look at slime glands, shall we? Little short videos are good for that kind of thing. This is a pretty effective defense mechanism.
Okay, for all of us.
That's when the ads are helpful. All right, that's a hack. They're so attractive, isn't it? Swim around, do his thing. So he's in the tank, and this guy starts to mess with him. And he's got slime glands throughout his skin that starts producing that stuff. Oh, that's so cool. And it just keeps making it. So the more this guy messes with that fish, the more slime this thing produces. That is an impressive amount of slime, is it not? How is that an effective strategy against predation, do you think? Yeah, it could clog up your gills. What else? Hard to catch you. Also gross, right? I'm gonna go eat a hagfish and I get a mouthful of slime. I might be deterred. Might choose to go try to catch something else, maybe, right? Pretty effective. I wanted to show you guys one more thing about this video. My favorite part. Check this out. At the very end, look at this. This dude has a jar with labeled hagfish slime. I don't know what he's doing, but he's collecting that stuff. Hilarious. Everybody's got a thing, right? Everybody's got a specialty. All right, so those are hagfish. No cranium, but again, no vertebrae, but cranium. So they're craniates, but not vertebrates. The lampreys are where it gets a little bit more tricky. So most classifications will call lampreys vertebrates. Okay, so they're going to go ahead and give these guys credit because they have really simple vertebral elements. So this is a schematic of the notochord, which is running, remember, down the dorsal surface of this organism, down the back of the lamprey. You've got the dorsal hollow nerve cord. It's just behind that cartilaginous nerve notochord. And look at these little purple things. These are called lateral neural cartilages, okay? These are like baby vertebrae, okay? So when you look at your vertebral column and your spinal cord, you essentially have, your notochord persists, but it is only in your discs, in between your vertebrae, okay? So this guy basically has that same, what will become intervertebral discs down the line, and it's got that nerve cord, so the support and protection is there, and then you've got this. These are like the earliest evidence of vertebral bones coming out around and surrounding that spinal cord. So in your vertebrae, they're stacked on top of one another, but there are holes in the middle. And your spinal cord runs through that stack, right? So this is what you're starting to see is this, what will eventually become bony elements of your vertebrae starting to protect that dorsal hollow nerve cord. Because lampreys have that, that's why we say, okay, you can be a vertebrate. So they have these little early rudimentary vertebral elements. All right. So some people say they're not true vertebrates because that's not a vertebrae, but other people say it counts. All right. So there's no real right or wrong answer there, but what you want to know about this group is that they are definitely cranians. Okay, so there's no debate about that. Both hagfish and lampreys have a cranium. Okay. Lampreys have eyes. Hagfish have simple eyes. Okay, but no jaws. So you're seeing, what you're seeing here is that this is a sucking mouth, kind of like the stolex of the tapeworm that we looked at. It's basically a strategy to be an ectoparasite on other fishes. So they connect up to the skin and they have what's called a rasping tongue, which sounds fantastic, right? It's like a scraper. So they suck onto the outside of the fish and they can scrape away some skin until they can start getting blood in them. All right, so that's lampreys. Jawless fishes, right? Agnathans. Now we get to the jaw. Let's talk about the jaw for a few minutes. So gnathostomes is anything that has a jaw mouth. There's that stone again, like protostome, deuterostome. Now we're saying gnathostome, and remember, gnath means jaw, so that just means jaw mouth. All vertebrates that possess a jaw are gnathostomes. Do you possess a jaw? You do. That makes you a gnathostome. Okay? So we are in this group. We are in this clade. We are included in the monophyletic group of gnathostomes. Okay? Let's see. This trait first appears between 440 and 416 million years ago. So in the big scheme of things, jaws aren't that old. If you think about the age of Earth, right? 4.6 billion years. Photosynthesis, 3.5 billion years. Eukaryotes, 2.7 billion years. Like 400 million years ago is like a drop in the bucket, right? So jaws are relatively new. So we start talking about these innovations, like a new thing, it kind of is in terms of life, the history of life, okay? The earliest gnathostomes and amphibians and placoderms, you guys don't really need to know what they are. I did want to show you Deuterostomes just because it's cool. Anybody play Animal Crossing? Any Animal Crossing fans in here? No? You don't have to admit it. Maybe you are secretly. I don't know. My kids play Animal Crossing, and they're not like babies either. One of them in high school, so I wasn't like trying to defend you guys with Animal Crossing, the baby game. Anyway, there's Deuterostomes in that game, which I thought was kind of cool. So this guy is interesting. We think we know what he looked like, but they've never found any of the entire fossils. Mostly they just found the jaw. But what we know is that he was a giant placoderm. Placoderm means plate skin. So these guys were early, huge, heavy, armored fishes. Okay, long extinct, but that jaw, that's the trait. Okay, that's the really important thing here. The first group that we're looking at that is extant, that we see examples of, and I think we'll stop here because we're kind of getting close. Maybe we'll get to the next fish group. We'll see. Are the chondrichthyes. What does the root word ichthyes mean? Michael, I know you know this. Yeah, fish, right? What does an ichthyologist do? Studies fish. So just as chondrichthyes, chondro is the prefix that means cartilage. When you see the next group of ichthyes, osteichthyes, you guys have probably heard that term osteo before. Osteoporosis, osteoarthritis. What does osteo mean? Bone. So if you can remember this one, because it's more familiar, you can probably remember that the other one means cartilage. So chondro means cartilage, osteo means bone, ichthyes means fish. So you've got your agnathans, which are your jawless fish, no jaw, right? And then you've got chondrichthyes, cartilage fish. Then you've got osteichthyes, bony fish. So the chondrichthyes are an older lineage, so we're covering that first. But this is the first group where we see those jaws. Give me some reasons, hypothetical or otherwise, that a jaw is a good idea if you are an aquatic animal. What can you do? Hunt. if you can grab something else and not let go of it and pull it into your digestive tract, you can eat stuff that no one else can eat. That is a tremendous advantage because food is what? Energy, right? Nutrition. You can intake more energy. You can move quickly enough to catch something, ingest it, and take all of that energy in for yourself. You can get bigger. You can get faster, right? So this is an incredible transition in the diversity of vertebrates, is the evolution of the jaw. And it comes from those gill arches. Okay, so it's just a change in the gene expression of what those gill arches become. Some of them are retained, chondrichthyes or cartilaginous fish still have gills, but that first gill arch moves forward and gives rise to this movable jaw that allows you to hinge and catch food. Okay, so this is a big, big deal. And the first group we see it in are the Jawless or the cartilaginous fishes, which include sharks. So if you've ever seen the movie Jaws, or you know what the movie Jaws is about, what's it about? Anybody seen Jaws or heard of Jaws? What's it about? Sharks. So if you know that much, you can remember that sharks are cartilaginous fishes, okay, and chondrichthyes. So the jaws appear first in this group. Another thing, so this is a really special group for a couple of reasons. That you also see first in this group is two sets of paired appendages. What are appendages, you guys remind me? Yeah, arms and legs, right? In this case, do they have arms and legs? They don't, but they have pectoral and pelvic fins. Pectoral, shoulder, and chest, right? So if you ever do like pectoral flies at the gym, right? This is pectoral muscles. Pectoral means chest, and pelvic is pelvis, yes, where your legs are attached. So your pectoral appendages, guess what? Those are your arms, and your pelvic appendages are your legs, okay? So we have them, but they first appear in the chondrichthyes, and they are paired fins, pectoral fins and pelvic fins. Those are homologous, you guys. Gene pathways, skeletal elements, homologous to your arms and legs. Okay, so you are technically descended from the group that gave rise to pelvic and pectoral appendages. Okay, so this is another big deal. The other characteristic in this group is that their internal skeleton is cartilage. It's not bone, but it's calcium infused. So if you ever look into studying the development of bone tissue, in us, most of our bones start out as cartilage. And then during embryonic development, those cartilage cells are broken down and replaced with bone, and calcium is brought in to mineralize it and make it hard. That happens to some extent in sharks. So they're not squishy, okay? They're mineralized cartilage, but they're not fully bone. So that's why they're called chondrichthyes or cartilaginous fish. The last thing to know about this group, first jaws, second paired appendages, third cartilaginous skeleton. The fourth thing to know is it's not just sharks, it's also skates and rays, like stingrays. Okay, so that's what you need to know about this group. That's a good place to stop. Because the next thing we'll do is talk about bony fishes, and it seems like it would be a short discussion. It's fish with bones, yes? But there's much more to it than that, of course. Of course it is. It's bio, right? So we're going to talk about some subgroups within the Osteichthyes. We're going to talk about why you are a fish, or at least your ancestors are fishes. You're considered a member of this group. We'll talk about that. And we'll look at, going back and forth, the evolution of life moving onto land, vertebrate life moving onto land. Okay, so that's a good place to pick up next week on Monday.